Okay, great. Okay, so let's do it together. So in that case, I want you guys to be as active as possible on the chat, guessing the questions. Obviously, I know you might be trying to solve it, but please do send in your messages in the chat with the guesses. If you want it to be a little bit more like you're not sure you aren't ever to see your answer, then absolutely fine. Just send it privately through me on the Zoom messaging, which works as well. So I don't want anyone to be shy about it. Literally just guess, and you're supposed to be making mistakes at this point. That's completely normal, but it's best to have the mistakes now than to have them in your exam. So I would really, really recommend a lot of participation and guessing the question, etc. Okay, so we'll start off with question one of the paper. Question one is usually about cells. So just in terms of strategies for this paper, the questions at the beginning will be a lot easier, so you should be able to get through them quicker. And then the questions at the end will pertain to a little bit more difficult questions. So some of them are more difficult, some of them are quite easy. But they are going to be more difficult simply because the content that's being asked is going to be more difficult. Okay? So I'd recommend when you get into that exam paper, know that you've got 40 questions. Know that you have an hour to solve 40 questions, and therefore that leaves you 1.5 minutes per question. But you want to solve the first ones a little bit faster because some of them that have graphs, for example, like this one, will require a little bit more because you're reading a lot more and you're trying to interpret data. Okay, some of them you might not remember things, so you might kind of have to spend time remembering the content, which is fine. So I'd recommend trying to go through the beginning ones quickly. And of course, you're gonna have the opportunity to come back and go look through your answers again and double check. Okay, so I would say spend about a minute per question, and that will leave you 20 minutes to check your answers. But yeah, so this is the full paper. Yeah, I would, as soon as you get it, flip through it, just make sure that you have the correct amount of pages, that you have the correct amount of questions, that you have the correct paper, which there shouldn't be an issue, but anyways. And yeah, so we'll get started. So for question one, which cell component is found in eukaryotic cells, but not in prokaryotic cells? D, okay. Any other, yeah, guesses? We've got A. So to answer this question, we have to kind of remind ourselves of what eukaryotic cells and prokaryotic cells is. So a trick that I like to do is I like to put them into kind of organisms that I know. So example, bacteria, and for prokaryotic cells, and then for eukaryotic cells, and I know obviously no true organelles. That's kind of a distinguishing feature. So before I even look at the answers, I know that this is going to be a bacterial cell, something like that, as an example, no true organelles. And then for eukaryotic cells, what I like to do is I think, okay, true nucleus, and then organelles. And then I like to put both plant and animal cells in my head. Because if I put plant and animal cells, then I remember that actually things that are in a plant cell will still be in the eukaryotic cell category. Okay? So the answer here is indeed A. And the reason is because which cell component is found in eukaryotic cells but not in prokaryotic cells? As we've said, prokaryotic cells don't have true organelles, so they're definitely not going to have mitochondria for respiration. But I understand that D might have hinted to say, oh, a cell wall, and we're thinking cell wall is a plant cell. But remember that cell walls can happen in bacteria as well. Okay? So bacteria can also have cell wall. Which is why the answer here is A. Okay, let's keep going. The drawings are from two original papers that proposed a structure of the cell membrane. Now, this is not the original picture whatsoever. The original picture was removed for copyright reasons, but I just put this in because I feel like it would replace the picture that was there well enough. So for what reason is the Singer-Nicholson model, which is the model that we see up there, considered a better representation of the cell membrane than the Davison-Danieli model? So have a go at trying to answer into the chat. Okay, you've got C. Anyone else? Any guesses? C. Okay. Yes, absolutely. It's going to be C. If a model is better than another model, then it has to do something that that other model doesn't do. Okay, so A says it has extrinsic proteins, which yes, it will have extrinsic proteins in the Singer-Nicholson model. However, the Davison-Danieli model has it as well. It shows has the phospholipid bilayers arranged. Yes, it does. However, the Singer-Nicholson model and the Davison-Danieli model both show that. They just have different representations. It helps in the understanding of the fluidity of the cell membrane. Now, this is new to the Singer-Nicholson model because the Singer-Nicholson model proposed the intrinsic proteins, right? So these intrinsic proteins that we see here. And when it proposed those intrinsic proteins, it also kind of helped understand the fact that these phosphate heads are going to be hydrophilic and that the tails are going to be hydrophobic, okay? And the fact that we removed this big, thick protein layer from the top with the original model, with the Davison-Danieli model, that allowed us to basically think, okay, well, these membranes, these intrinsic proteins, they're intrinsic, so they're within the whole membrane, and they're free to kind of move around, and they just move around the membrane. So that's the fluidity part. So the intrinsic proteins now being embedded in the membrane led to the fact that the layer of proteins at the top now isn't restricting the membrane movement. And also because it made us think about this being hydrophilic, this being hydrophobic, and the same thing with the proteins. So the proteins themselves had to have hydrophobic ends and hydrophilic ends, okay? It shows how oxygen can diffuse into the cell. Not really. Again, that's not really something that's covered in the syllabus about the Singer-Nicholson model. Yes, oxygen can diffuse in the cell, but that's not really the advantage of the Singer-Nicholson model. Okay, cool. And it wasn't the purpose of it either. So the diagram shows the protein channels involved in the passive movement of a substance into the cell across the membrane. So substance going into the cell. What describes this movement? Okay, so we're on question 3. Anyone have any guesses? D? Yep, yep. Nice. Okay, so let's have a look and let's see if we can describe it the best we can. So as we can see, the things when we want to look at transport for is, first of all, the first thing I'm looking at is it following the concentration gradient? Because when I look at is it following the concentration gradient, that allows me to establish if it's passive or not passive. Okay, so is it following the concentration gradient? Is it going from an area of high concentration to an area of low concentration? And in fact, it is. So I'm looking at a passive transport. It even tells me here that it's a passive movement of a substance. So if it's a passive movement of substance, then therefore it's definitely not going to go from areas of low to areas of high concentration. And I even double checked that by looking at that in my question. So it's definitely not going to be B. The next thing I'm going to look at, is there any molecules that look like they could be representations of ATP to show that it could be an active transport? And again, I don't have that. They're just kind of flowing through. The only thing that could kind of trip me up is the fact that it's going through membrane protein. And if you forget, this can be tricky because you're thinking, oh, a membrane protein that has to use ATP. But no, remember that we have membrane proteins and channel proteins. Both are involved in ATP and passive transport. So the mechanism shown here by this image is known as facilitated diffusion because it's passive, but these molecules are big, so they require a passageway. They require something, a protein, to help them get across the membrane, either because they're big or because they're charged. So here we can definitely see that this is facilitated diffusion because there's no molecule like ATP being used, and it is following the concentration gradient. So just because there's a membrane protein there doesn't mean the fact that it's ATP, because it's only when it's a pump. So there's only one specific membrane protein that will need ATP. This one will not. Okay, so then A can be excluded. The proteins ensure the movement of substances only in one direction. So yes, there is arrows pointing to go this way, but it's not the proteins that are ensuring that. It's just the concentration gradient. So no. Net movement occurs until the concentration in and out of the cell are equal. This is the only option left, and this is indeed a true statement that it will happen until both sides of your cell, so both sides of your membrane, your intracellular and your extracellular space, are equal. And then the net movement will stop. So there might still be transfer back and forth, but the net movement overall is going to be 0. Okay? What is the evidence for the endosymbiotic theory? So we've got 4 options. Please type into the chat what you think. Like I said, the best way to, practice is to try and solve questions. Okay, so we've got A's. Okay. Okay. So let's have a look at the evidence for the endosymbiotic theory. So a really good thing is before I look at these options, I want to memorize, I kind of want to revise in my head the content that I know. And I feel like that really, really helps. So evidence for the endosymbiotic theory, I remember there was four things. There was the fact that it was in the mitochondria, first of all, and the chloroplast. And what are the four things that the mitochondria and the chloroplast had that proved the endosymbiotic theory? Well, they had double membrane around them, which means that one membrane was from the original cell, was from the original prokaryotic cell. The other was from the engulfing. The other thing was they had ribosomes that were 70S, which is common in prokaryotic cells. They divide via binary fission. They also have a naked ring of DNA. And they had plasmids as well, which is all common in a prokaryotic cell. Sorry, I meant to write plasmid, not DNA again. So for this reason, so these are the four reasons. So even before I'm looking at my answers, I'm kind of in my head like thinking, okay, cool. Let me look at options A, B, C, and D. So A says eukaryotic mitochondria contain DNA. Yes, that could be one of those, right? B, prokaryotes evolved before eukaryotes. That's not the evidence for the endosymbiotic theory, because there's evidence for the endosymbiotic theory, and that is four specific things, right? So that's not part of those four specific things. Unicellular organisms exist as both prokaryotes and eukaryotes. Again, that's not really evidence for the endosymbiotic theory. That's just telling you that eukaryotes can be unicellular as well, and so can prokaryotes. That's not really evidence for an engulfment and a symbiotic relationship. Prokaryotic cells have no double membranes. So a normal bacterial cell will not have a double membrane, absolutely yes, but that's not proof. So if I were trying to prove that one cell was engulfed by another, the fact that a normal prokaryotic cell only has one membrane, while the mitochondria inside has two, that doesn't show anything, that doesn't say anything. However, if I'm thinking, okay, this mitochondria has two, the mitochondria and the chloroplast have two membranes, that means that they were engulfed by the eukaryotic cell's own cell membrane as well. So the fact that the prokaryotic cells in general have no double membranes, that doesn't prove this theory. What proves this theory is that the organelles themselves have a double membrane. Okay, hopefully that makes sense. That's why D is not true, and the answer would be A. Okay. Next, the image shows a cell from the root tip of an onion during late prophase of mitosis. How many chromatids are present in the cell? Now, of course, you can kind of count it, but I would not recommend doing that because they're a little bit overlapping. It's a bit hard to count. So what could we do instead of counting? Well, we could just look at the number that's given to us. The diploid number is 16, right? So what would the answer be here, guys? So we've got an answer that is D, okay? So the diploid number is 16, which means that during late prophase, remember the cell cycle stages? So we have interphase that happens through G1 phase, and then we've got S phase where the cell division takes place, and then we have G2 phase, which is the growth 2 phase, and then we have PMAT, so prophase, metaphase, anaphase, telophase, and then we have cytokinesis. So these are the cell cycle stages. As you can see, by the time you get to prophase, you've already gone through S phase, so you have already duplicated the DNA. Now, the diploid number for an organism, so the 2n number, is the total number of chromosomes in that organism. So the total number of chromosomes in that organism. And so if the total number of chromosomes in that organism are 16, when a cell is dividing and has gone through G1 and S phase specifically, where the DNA replication happens, then that organism has duplicated its genome. So it's gone from having a diploid number to having double the diploid number. So the diploid number, which is 2n, represented here, times 2. because that's what happens during the S phase, right? That happens during the DNA replication phase. And so 2n times 2, that's your diploid number. That is what happens during S phase. Now, can we check that our root onion tip has gone through S phase? Yes, we can, because it says it's already in late prophase. So it's already at prophase. So it's passed through G1, S phase, and G2 phase already. So it's already duplicated the DNA, and now it has them in chromosomes, right? So how many chromatids, however, are placed in the cell? Well, like we said, we're gonna have the diploid number, which is 16 times 2, right? And that diploid number, which is 16 times 2, refers to the chromosomes, right? And these chromosomes are made up of, so this is a homologous chromosome where you have the maternal copy and the paternal copy. That's usually how it looks like in your cells, right? But when we say that we are duplicating the DNA in a root of an onion tip, so even though these chromosomes have maternal and paternal copy, when we duplicate them, then the number of chromatids is different, right? So the number of chromosomes and chromatids are different in the cell. And what we can deduce here, because it's a diploid number of 16, because the diploid number is 16, that means I have 16, so I have 16 total chromosomes, which means I have basically 8 homologous pairs, usually in the organism. Okay, so the 16 is referring to the total number of chromosomes, which is referring to the fact that we'll have 8 pairs, which gives me 16 chromosomes in total. Okay, so it's kind of like in humans, we have 46 chromosomes. Our diploid number is 46, but that means I have 22 pairs plus 1 sex chromosome. So I'll have 23 pairs. So here I have 16 chromatids is basically, it's basically, in summary, that's what it's saying. The 16 is how many overall chromatids, but I will have 8 chromosome pairs. Is everyone following with that so far? So the diploid number is the total number of chromosomes, but it means the whole number of your chromosomes, so all of your chromatids, if that makes sense. Whereas it doesn't show you the number of pairs of chromosomes. Okay, so like we said, the diploid number in humans is 46. Doesn't mean I have a total of 92 chromosomes. It means I actually have 46 chromatids, and they're paired up in pairs of 22 plus one pair of sex chromosomes. Does that make sense? So the 16 gives me the indication of the number of chromatids in this diploid number. The 16 then gets duplicated, which gives me 32 chromatids during S phase. Does that make sense? But I think I understand what the confusion was, because the confusion was thinking like 16, so that means I'll have 32 chromosomes, which means I'll have times 2 of that chromatids. But I think it's more that the 16 is the chromatids number. Just like in humans, we have 46 chromosomes, but technically 23 pairs. Yeah? If that makes sense, I'm going to move on. But let me know if it doesn't make sense, please. I don't mind like redrawing or like re-explaining it a little bit, because this is kind of a really common question. And I'd rather you guys get it correct in your tests. Okay, makes sense? Okay, cool. So the answer here would be C, because remember that when it says like 16, that means 16 overall chromosomes, which means 8 pairs of chromosomes. Okay, so the 16 is actually referring to the number of technically chromatids, just like in humans. So 1, 2, 3, 4, 5, 6, 7, 8. So this is how much chromosomes we have. We'll duplicate that. So we'll duplicate that times 2. That will give me 32. And obviously you can count it up. Cool. So next question, which drawing represents beta-D glucose? Remember those things that you just have to memorize, like the structure of beta glucose? This is one of those. So exactly. So you should be able to answer this immediately. So this should take you no time, right? Because you remember that beta glucose has the OH group on the top, which is exactly here. So that's A. Okay. Cool. What is the proteome of an individual? The amino acids unique to an individual making up proteins in the cell. The way in which an individual's polypeptides are folded into a 3D structure. the protein synthesized as an expression of an individual's genes, all the possible combinations of an amino acid an individual contains. Let's have a look. So what is the proteome of an individual? So if I remember my definition of the proteome, it's all the proteins that are made from the genome. Because if you guys remember that slide in the crash course, the proteins that are going to be made, they're being made from the genome. So the genome leads to the proteome, right? So let's have a look at these options. So the option A says the amino acids, so the amino acids unique to the individual make up the proteins in the cell. So the reason why this one I would kind of exclude is because it says the amino acids, and the proteome really is, yes, it's amino acids included, but it's really more proteins, right? The way in which an individual's polypeptides are folded into a 3D structure, no, that's just protein structure, right? It's not actually the proteome. The protein synthesized as an expression of an individual's genes, yes, because it's the proteome being dependent on the genome. So it's all of the different combinations of proteins that that person could make. So not necessarily that they're making right now, but what they could make. Because let's say in a cell at this point in time, they're not using this function or they're not undergoing this function. That doesn't mean that that protein that's not being used is not part of the proteome, right? The proteome is anything that the person could make depending on the genome. I remember what we said with that slide. On that slide on the crash course, we said the proteome gets more complex as you add more post-translational modifications, as you keep adding modifications to the amino acids, etc., etc. Okay, so that will be the proteome. So that's why option C is correct. All possible combinations of amino acids, again, it's not amino acids, it's going to be proteins, so we can exclude D. Okay, cool. In the grass plant Hylopyrum mucronatum, the enzyme amylase breaks bonds in polysaccharides during germination. The graph shows how the activity of the enzyme varies with the concentration of polysaccharide. Just off the bat, I can see the fact that the concentration of the polysaccharide, as it's increasing, the rate of reaction is increasing as well. So I can see that there, and I can see that there's somewhat of a plateau in the data points, not in the trend line, but in the data points, right? What is the reason for the curve labeling off? So that's the fact that it's going to plateau. So sometimes they will use different words to what you might see in class. Always just remember, what does this mean? Plateau. Okay, fine, plateau. That might help me remember some things if I forget. So just put it into your own words. There is insufficient substrate for the enzyme to act on. Well, what is the substrate in this case? The substrate is going to be your polysaccharide. So that's going to be your y-axis. And B says it's going to be insufficient. So, I mean, A says it's insufficient substrate. I don't think it's insufficient because it's increasing in concentration, right? So it's not going to be A. The product acts as an enzyme inhibitor. So what's the product? The product is probably a sugar. Is it inhibiting the enzyme? Do I see a curve that looks like enzyme inhibition? Not really. I just see the fact that the enzyme active sites are getting saturated, right? C says the enzymes have all been consumed. No, otherwise they wouldn't be still working at this very high rate of reaction, right? So they're still working really hard. It's just that the actual rate is not getting any faster, okay? And so the reason for that is the enzyme sites are occupied by substrate already. So absolutely that's D. So if this is something that you kind of struggled with, just remember the labeling of the curves and the different reasons for the increase. So the reason why the curve initially increases is because of the fact that you have a lot of active sites and they're not all being used up. So they're not being used to full potential. So you have, like, for example, 3 active sites being used up, right? And so that gives me, I don't know, rate of 3 polysaccharides being broken down, for example, per minute, something like that. Now let's say I add more substrate. Now I have 4 enzymes working. So therefore that will increase my rate to 4 polysaccharides per minute, something like that. Then let's say I have all of them working, so I've got 5. That will increase my rate again to 5 polysaccharides per minute, right? So I'm increasing my rate throughout this whole time. Now let's say I add another substrate, another substrate, another substrate. It doesn't matter because all the active sites are already being used up. They're already saturated. So absolutely the answer would be D here. Okay. The anticodons of the 3 tRNAs in the amino acids they carry are shown on the table. Which base sequence of an mRNA molecule would code for arginine, leucine, valine tripeptide? So try and answer this question, and I will reveal the answer. C, okay. D, okay, D. Okay, cool. Let's have a look. So here we have the tRNA anticodon. So the tRNA anticodon, it's the anticodon, so it's going to be the opposite of what's shown on our mRNA molecule. Okay, so first, before I try to figure out the opposite of what's here, I want to reorder my amino acids in the order that they've asked me in the question. So they want it to be arginine, so they want it to be GCA first, then they want it to be leucine, which is going to be AAU on my tRNA, and then valine, which is going to be CAG. Okay, so if this is my tRNA molecule, again, I love drawing things on an exam paper because it means that I'm visualizing, that I'm understanding, that I can really apply my knowledge. This is all my tRNAs. Then let's have my mRNA. So how would my mRNA look like? Well, my mRNA would be the exact opposite of that. So it's going to start with C, G, and then instead of it being T, it's going to be U. So we don't have T's yet because not it's the DNA. It's still an RNA strand. So we can already exclude B and C because they have T's in them. We know that they definitely can't be that because that will only be in a DNA strand. But we're looking at mRNA. So then next up it will be U, U, A. Then it will have G, U, C. And which one of them matches this perfectly? Well, it will be D. And so therefore the answer is indeed D. 10. Which graph represents the action spectrum for a green plant receiving only blue light? Any guesses? B? Okay. Anyone else for B? No? Okay, cool. So the answer is indeed B. So how do we figure this out? Well, it's asking for the action spectrum, and I have to dig deep in my memory and think, okay, what was the action spectrum? Oh yeah, it was the wavelengths of light that were used up in photosynthesis. And what does the normal one look like? Well, the normal one looks like this, and the reason it looks like that normally is because you have your green wavelengths that are not being used up, so this is definitely green. But I'm trying to kind of remember which one is going to be blue. Is it going to be this curve or is it going to be this curve, right? So remember the relationship between energy and wavelength. So higher energy is actually going to be your shorter wavelength. And red light is lowest in energy versus the blue light, which is the highest in energy on the spectrum. So if you remember your electromagnetic spectrum, it goes like purple, which is the highest, goes blue, and then it goes to greens, and then yellows and reds. So that's the way kind of order it goes in terms of high energy to low energy. So we're looking for something that will only absorb blue light. So it's going to only have high energy. And so the high energy is inversely proportional, or like the exact opposite to the wavelength. And so high energy equals small wavelength. And so this curve is going to be my blue. This curve is going to be my red. But of course, if you've just remembered how it looks like, then you don't really need to do all of the deduction. But I'm just doing this deduction in case you completely forget. The other thing is if you can't even do this deduction, as long as you remember the first one looks like this, you can still answer the question. Because if you thought this was, for example, the blue wavelength, well, that doesn't exist here, right? So we don't have an option that's just this. We have an option that's C, which is showing us the green. So this is never going to be absorbed. And we have an option that's showing us just reduced use of both sets of colors. So is it blues or reds? Both of them are being used less, so it won't be this one. And so by process of elimination, I know it's going to be B because I know it can't be my original one because my original one shows how we use both blues and red lights. Okay, cool. What change causes sickle cell anemia? So this is one of those questions that like you should be able to answer without looking at the options. So what change causes sickle cell anemia? Before I even look, I'm thinking, okay, base substitution mutation. So you're getting C's. Yeah, perfect. And I know that it goes from GAG to GTG. So before I even look, I know this is the thing. And then I can go back to my answers and say, which one doesn't match this? One amino acid less? Nope. A mutation leading to an extra codon? No. Thymine replacing adenine? Oh, sorry, guys. Thymine replacing adenine? Yes. Failure of tRNA to correctly transcribe the sequence? No, it's a full base substitution. So a whole base is substituted during the DNA synthesis, during the DNA replication, during the transcript, during the actual synthesis. So that's why it's going to be C, because it's a genetic mutation. And so therefore we can't have D. So definitely the answer is C, like you guys get, which is perfect. At what stage of meiosis do chromosomes pairing and crossing over occur? B. Yep, absolutely. Yes, so remember that crossing over will occur during prophase 1, and that's where they exchange the genetic material and they form the recombinant chromatids. Okay, interphase definitely not, because that doesn't happen in interphase. Interphase just is growth phase, S phase, and G2 phase. Metaphase no, because they're lining up along the plane in metaphase. Prophase 2, it doesn't happen in prophase 2 because we don't have the whole homologous chromosome pairs. We don't have the tetrad anymore in prophase 2. We just have that only in prophase 1. So that's why it's not going to be prophase 2, because in prophase 2 we just have these two, just the two chromosomes together. But in prophase 1, we have all 4 of them together. Okay? The pedigree chart shows the inheritance of 3 generations of Duane syndrome, a condition caused by a dominant allele that affects the alignment of the eyes. In individuals II, that will be this individual, and so if individuals II1 and II2, so these 2 individuals here, had a third child, what is the probability the child will have Duane syndrome? So have a go at trying to guess it. B. Yep. Getting a B there. Anyone else for another answer or the same answer? Okay, so the answer is indeed B. So let's have a look at why that is so. So with these pedigree charts and trying to find the probability, trying to find the probability the child would have Duane syndrome, if it kind of doesn't come intuitive to you, then you always can rely on the method of using a Punnett square. So how would you do a Punnett grid or a Punnett square? Well, you have to know the parents' genotypes, and then you have to cross them over, right? So the parent's genotype, II1, well, it's a dominant allele, so therefore this person has to be recessive homozygous, so that person has to be, because they're unaffected, so it has to be, for example, small d, small d. This person can either be big D, or they can be big D, small d, right? But, however, if they were big D and big D, they would have to get both dominant alleles from both parents. And I can see that this parent definitely has a dominant allele, but this parent definitely does not have a dominant allele. She definitely has a recessive genotype because she's not affected. And so therefore we can't have big D, because she would only be able to get the dominant allele from one parent, so she will be heterozygous. So then we have our Punnett grid, which we have small d for one parent, big D, small d for another parent, and then we can cross. And therefore you would get the chance that the child is affected or would have Duane syndrome is 2 out of 4, which is about 1 out of 2, which is 50%. And so the answer would be B. Okay? So another thing that you could use, a trick to kind of make it intuitively come to you, is if you know that the mom is heterozygous, then you know the mom has a 50-50% chance of passing it on. because she can either give the child this allele, or she can give the child the other allele, right? So it's 50-50, okay? But that only comes after deducing the fact and after confirming that she is in fact heterozygous, because she might be homozygous, so don't forget that. You just have to look at the parents first to be able to check if she is or not. Okay, cool. What is combined in the production of cloned embryos by somatic cell nuclear transfer? So we've got an A. Anyone else? Yeah, A. Exactly. That is in fact the correct answer. Because in somatic cell nuclear transfer, what we have is we have a somatic body cell. So we take a cell from the thing we want to clone, so thing that needs cloning. So for example, if it's a cow, and we take that normal body cell, we remove as we take the normal body cell, we remove the nucleus from that cell because we want the DNA of this thing that we want to clone, and we move it into an egg cell. And this egg cell will be taken from a donor. And this egg cell has all of the nice nutrients for the new embryo to grow after fertilization and everything. So it has all the mitochondria, it's ready, it's got everything. It's like a nice home for the new embryo or the new zygote. And so we're gonna basically take the original nucleus from this egg cell. We are gonna subject it to UV light therapy. So we're gonna actually destroy it via UV light. So we're going to destroy this egg cell's original nucleus. We're going to remove it from the cell. Okay, so we are going to completely remove it. And then we are going to transfer this nucleus into this egg cell. And that's how somatic cell nuclear transfer is going to work. And then afterwards, we take this, we fertilize with sperm, and then we transport it into the organism or the surrogate organism. So we inject it into the surrogate organism. Okay, so that's how somatic cell nuclear transfer works. So let's see what option matches that process the best. So an egg cell without a nucleus and a nucleus of a somatic cell, yes, that does match it because we take the egg cell that's without the nucleus from the donor and the nucleus of a somatic cell that we've transferred. And we can see that red nucleus that's been transferred here. An egg cell nucleus, are we taking the nucleus of the egg cell? No, we're taking the nucleus of the somatic cell. And the somatic cell has to have the nucleus because the somatic cell is the body cell from the organism that needs cloning. An egg cell with a nucleus, again, no, we're not taking the egg cell's nucleus. We're taking the somatic cell's nucleus. And a somatic cell with a nucleus, no, we're not going to have two nucleuses. An egg cell nucleus and a somatic cell nucleus, again, no. So therefore, B, C, and D are incorrect. Okay? Which statement applies to transfers in an ecosystem? So transfers refers to energy transfers, carbon dioxide transfers, heat transfer, all those three different things. So we're going to have to check which one is correct. So statement A says, green plants can transfer heat energy from the sun into chemical energy. So they're saying that the UV light or the energy from the sun, the inorganic energy, is being transferred from the sun into chemical energy. And while plants definitely take energy from the sun and transport that into chemical energy, that's definitely what photosynthesizing plants do. It's not heat energy, it's light energy. And so that's why A is incorrect. The greatest loss of energy occurs towards the end of the food chain. So the loss of energy is the transfer of energy between different trophic levels. And in fact, the energy that's lost between every single trophic level is exactly the same. So only 10% of energy is passed on, which means you're losing at every single trophic level 90%, minus 90%, minus 90%. So there's no actual greatest loss of energy anywhere in your different food chain, anywhere between your trophic levels. So it won't be B either. Production of carbon dioxide by respiration results in loss of biomass in the food chain. And this is true because in cell respiration, we know that we take the biomass. So let's say we have a consumer here. The consumer is going to consume biomass. So for example, let's say we have a rabbit and the rabbit is eating grass. So he's consuming the grass, he's consuming the biomass. And what he's doing is he's taking apart the molecules in the grass, maybe grass was a bad example. Let me say carrots, because carrots actually have sugars. And he's taking the molecules in the carrots that he's consuming and he's using them up. He's taking the glucose from the carrots, he's taking the starches from the carrots, he's using it up and breaking those sugars down to then do cell respiration and release CO2. Okay? So via cell respiration, you are taking biomass, so other organisms, other plants, you are breaking them down into you're breaking their sugar molecules down, you're breaking all the nutrients down, fats, everything, and you're releasing CO2. So production of carbon dioxide via respiration, which is cell respiration, results in the loss of biomass in a food chain. So absolutely that is true. What about D? Both energy and nutrients are finite and must be recycled? No. Nutrients, yes, will be recycled. Energy cannot get recycled. Energy is just lost to the environment. And so that's why the answer is C. Okay, does that make sense? If it doesn't, by the way, just let me know. I don't mind re-explaining it a different way. Okay. Next question is question 16. How do methanogenic archeans living in peat bogs obtain energy to survive? So this one's a bit of a tricky question because there's a lot of words, but have a go at trying to answer it. Okay, perfect. So that answer is in fact B. So let's have a look at these different options. So first of all, what do methanogenic archeans do? They produce methane. Okay, we spoke about that in the crash course. We talked about how they produce methane. And they produce methane when they do this in waterlogged conditions. And that's because the waterlogged condition creates an anaerobic environment where there's no oxygen and these waterlogged, so the reason why, well, waterlogged basically means like blocking of where the soil is not able to drain water. So the water being there compacts down and basically like fills up all the soil so that soil doesn't have any chance to gain any air. And so that's what waterlogged means. And that's why it creates an anaerobic environment because now you don't have any oxygen coming in from the atmosphere. Okay? Anaerobic and also acidic environment as well, right? So that's how peat is formed from, or that is, sorry, how methane is formed from these methanogenic archeans. So let's see what option matches that description the best. They oxidize methane produced by peat. They don't oxidize methane, they produce methane. So that one's not correct. And they don't produce CO2 and water, they produce methane. That's why they're called methanogenic archeans. So another reason why the option is incorrect. B, they synthesize methane. Yes, they do, from carbon dioxide released by decomposing peat. Yeah. They convert organic compounds in a fully decomposed vegetation. No, because the decomposing peat is organic material that's only partially decomposed. Right? And that's the whole point, is you're decomposing things that are partially decomposed. And so this one is incorrect because we don't use fully decomposed vegetation. D, they produce methane while converting peat to coal. No, who said anything about coal? This is definitely not part of definitely not part of the correct answer. So the answer is B, which is an example of evolution by selective breeding. So let's go through question 17. B, yep. So selective breeding is a form of artificial selection. So it's not selection by nature. So with any kind of question like this, you want to look at what things could we have humans selected for? What would a human had selected for? So selection of prey animals that can run faster than the predators. So seems like it's a natural selection because we haven't selected for prey animals to run faster than predators. We haven't done that. That's not something that humans have done. So that seems more like natural selection. The variation in size of the different breeds of dogs. Yes, that is something that we have naturally selected for. So humans like to have, we have artificially selected for. Humans like to have smaller dogs. Sometimes they like bigger dogs. So the different breeds of dogs, especially whenever it mentions breeds of dogs, think, okay, this could be definitely a viable answer because it could be artificial selection here because it's breeds of dogs. So that can apply to, for example, how tame a dog is, how some dogs don't even bark and they've been selected for that because some people don't really like dog barks, so they want a dog but they don't want it to bark. So they will select for a dog that doesn't bark and therefore they will have that dog. So breeds of dogs having different characteristics, that's the thing that a human would select for. Okay? C says the tendency during breeding for birds to produce more offspring than will survive. That's part of natural selection. That's the overproduction of offspring. No, so it's not C. Some female spiders only breeding with males, which makes the right signals. Female spiders being able to select their males, that's also natural selection. So the answer is B. 18. Which statement best describes how evolution occurs? We've got a B in the chat. Anyone else have any guesses? Oh, C. Sorry. Okay, C. And that's why I have a go. Question 18. Here we got C as well. Absolutely. I think everyone's kind of on the same page with this one. So how does evolution occur? Well, evolution is, natural selection, survival of the fittest. So we're thinking about that. Let's go through each of the options and do process of elimination, right? So A says species which produce the most offspring are favored by natural selection. No, that's not true. There is something in there about species overproducing offspring, and that means that the resources are limited. But they're not favored by natural selection because they're producing the most offspring. Mutations in somatic cells are passed on to offspring. So somatic cells, remember, they are the body cells. So they're an opposite to a different type of cells known as the gamete cells or the sex cells. So these are opposite to that somatic cell. So mutations in somatic cells are not passed on to offspring. Okay, because the mutations that are passed on to offspring are ones that are going to occur in the gametes, which are your sperm and egg cells, so your sex cells. Okay, so this one's absolutely incorrect. C is the correct answer because natural selection does decrease the frequency of unfavorable characteristics. So characteristics that are not fit to survive the environment, natural selection will make it so they will struggle to survive too much and therefore they will not survive and produce offspring. Change in species lead towards greater complexity over time. Not really. You can have same complexity over time, or you could have decreased complexity over time. Natural selection has nothing to do with like complexity of a species. Okay. What occurs during inspiration? You got a D. So two things we have to think about here with inspiration. We have to think about, first of all, what pairs of muscles are contracting, and then what's happening to the pressure in the thorax. So the thorax is the chest cavity. So it's your actual chest. Anyone have any other guesses? See? Okay, so let's go through it. So like I said, two things we have to think about. So first of all, we have to think about what muscles are contracting. Is it the internal intercostal muscles or the external intercostal muscles? So if you remember the internal and the external intercostal muscles, they're the muscles in between your rib cage. So if this is my rib cage like so, I have muscles in between those ribs, and these are called your intercostal muscles. Okay, so we've got both sets of internal intercostal muscles and external intercostal muscles. Okay, so if you remember from the crash course, we said that the ones that contract are the opposite of what's happening. So what does that mean? Well, this is inspiration. You'd say, okay, inspiration, internal intercostal muscles. No, it's going to be the external intercostal muscles. So think whenever it's inspiration, it's going to be external. Whenever it's expiration, then it's going to be the internal intercostal muscles. So first part of the question addressed, we can remove both A and B because it's not going to be the intercostal muscles. Now, what about the pressure in the thorax? So if you remember, pressure is inversely proportional to volume. So the higher the pressure, the less the volume. Okay? So let's have a look at what's happening when you breathe in. So when you breathe in, can you guys feel the volume in your chest expanding? When you breathe in, you are adding air in, so you're actually making the volume of your chest bigger, right? So you're increase the volume, which means you're going to have less pressure. Okay, so that's why the correct answer is C, because you don't increase the pressure in the thorax, you actually decrease it because of the volume. So you can physically feel your chest expanding. And it's not just an expansion thing, but also the fact that your diaphragm, when you breathe in, is going to contract so that it makes more space, more volume in your chest. So think about the space and the volume. Okay, so during inspiration, your chest cavity will get bigger, your thorax will get bigger. And the reason why is because you have to allow more air in. And so that's going to decrease the pressure in the thorax, right? During expiration, your cavity gets smaller, your thorax gets smaller because you want to push the air out. Okay? So that's why the answer would be C. The diagram represents transmission across a cholinergic synapse. So what does cholinergic synapse mean? It's just a synapse that uses acetylcholine as the neurotransmitter. Where would a neonicotinoid pesticide act to prevent synaptic transmission? So have a go at trying to answer this one. So we've got D. Yeah, okay. Yeah, perfect. Okay, so let's go through it. So anyone choosing A? Hopefully not. So this is the mitochondria. So this will be important to make the ATP for the neuronal transmission, for the action potential, but definitely not where a neurotransmitter would bind. C is the vesicle. So this is the vesicle which contains the neurotransmitter. We call the synaptic vesicle, or where the neurotransmitter is contained. So a neonicotinoid pesticide prevents synaptic transmission. So the pesticide could bind to C, so due to the neurotransmitter directly, because that's your acetylcholine. However, it needs to bind to C and inactivate it, basically. However, if you remember from the crash course, we talked about how these neonicotinoid pesticides, they're going to bind to the receptor and prevent the binding of the acetylcholine. That's how they work. So we can exclude A and B kind of easily, but between C and D, it can be tricky if you don't know the mechanism of action of these neonicotinoids, these pesticides. And in fact, what they do is they bind to the receptor, and therefore they prevent the binding of the neurotransmitter. Okay? And so that's why D is the correct option. Okay, so we can remove that as well. For what reasons are daily FSH injections given during IVF treatment? So have a go at trying to answer this one. We have passed the halfway mark, so we're nearly there. Okay, we've got B. Yep, so indeed, so remember the different steps of IVF. Number one was superovulation, and that's the one that we used the FSH in. So we give huge amounts of FSH. Oh, sorry. Step number one was downregulation. And then downregulation, we used all of these hormone blockers. Right, so the nasal spray, and it's just formed of a lot of hormone blockers to prevent ovulation, right, to start like a clean slate. Then step number two is superovulation, where after we selected a few eggs, we give them a lot of FSH, and the role of that was to ovulate extra, so to produce more eggs than normal. And even if this is not something that you remember, just think about what the function of FSH is. FSH is follicle stimulating hormone, so it's going to stimulate the follicle, the egg. And so superovulation FSH is going to stimulate eggs. So from these options, we have to choose an option that's going to be related to FSH doing that. So is it going to suppress the natural menstrual cycle? No, that's what your hormone blockers are for in the first step. To induce the ovary to produce more eggs than normal? Yes, because this FSH has a function of stimulating the follicle to produce eggs, to help eggs to mature. To prepare the lining to the endometrium? No, that's what progesterone is there for. That's what gets injected for. Did it prevent the development of multiple embryos? No, we don't really have anything against that. It's just that's not part of the IVF process. So it's going to be B. So the image shows an X-ray diffraction photograph produced by Rosalind Franklin. What did Watson and Crick deduce about DNA from this image? B. Exactly, the DNA molecule is helical in shape, because it was one of those images that's so characteristic and so famous in science that this kind of answer should come very easily to you, because Watson and Crick, remember, they found this image, this was Rosalind Franklin's image, and they found it, and then they took the credit for deducing that the DNA was a double helix shape, right? But in fact, no, it wasn't a double helix shape. Sorry, in fact, yes, it was a double helix shape, but in fact, no, it wasn't their work. DNA carries the genetic code. No, we'd already known that, and that was from the Hershey and Chase experiment. DNA has 2 strands of nucleotides linked by bases. So, yes, this was discovered, but it wasn't discovered from this specific image. DNA is composed of an equal number of purines, A, G, and pyrimidines, C and T. So, indeed, this was discovered as well, but it wasn't from this picture. This picture, specifically from this image, X-ray crystallography, was the DNA molecule is helical in shape. Okay, so moving on. 23. What happens to an RNA molecule in eukaryotes after transcription in order to process it into mRNA? So this is the post-transcriptional modifications from topic 7. So we've got AC. Cool. So remember, in transcription, after it, so when you have your immature mRNA, you have a bunch of introns and you have a bunch of exons. And before you mature your mRNA, you first have to remove the part of it that's not being coded for. So the part of the DNA that doesn't code for anything. And so we're going to remove the part of the DNA that doesn't code for anything. So which is that? Is that the intron or the exon? So if this is my intron and this is my exon, for example, what am I going to remove? Well, I have to remove the part that doesn't code for a protein. And remember, exons, they are expressed. So exon, EX, exon is expressed. And so these will stay and the introns will be removed. Okay, because they're not expressed, they're not forming the DNA that codes for a protein. So therefore, A is incorrect, and so is B, because exons stay, they're not being removed. Then option C says adenine nucleotides are added to the 3' end, and then adenine nucleotides are removed from the 3' end. So remember, after the exons are expressed and after removal of the introns, the next step is to add the poly A tail, which is where you go to the 3' end of your mRNA molecule, and you add a bunch of A. You add about 150 to 200 A's at the 3' end of your mRNA molecule. That was the second modification. The third modification, if you remember, is adding on your mRNA molecule, so on the other side, 5-methyl capping. So it's called 5-methyl capping because it happens at the 5' end of the mRNA. So if I have a bunch of A's on the 3' end, and this is my 5' end, I'm going to add a methyl group. And this is called 5-methyl capping. So these are the 3 different post-translational modifications you need to be aware of. So that's what the question here is asking. So indeed, we're adding adenine nucleotides to the 3' end because we're not removing them. We're adding a bunch of AAA in our poly A tail at the 3' end. Okay? Cool. Next question, 24. So the original image was removed for copyright reasons. So and it's asking about bacteria rhodopsin, which is a protein in a bacterial membrane. I actually found the image of a bacteria rhodopsin. This is not from the original paper whatsoever, but it just shows you what you need to know to be able to answer the question. So I thought we could practice this skill as well. So bacteria rhodopsin is a membrane protein in bacteria that acts as a protein pump. Its structure consists of a single polypeptide strand. So it's one polypeptide strand, but it folds a lot of different ways. What is the highest level of protein structure from this image? So I'll give you guys a couple more seconds to answer. Okay, so let's go through it step by step. So A is primary. So primary means that it's going to be only an polypeptide chain. So just going to be amino acid, amino acid, amino acid. And while this question shows us that it's going to be a single polypeptide strand, that doesn't mean that there are no interactions between that polypeptide strand. So this is a primary is with no interactions. So no types of bonding, no special folding, nothing like that. So this is just an amino acid chain, just a straight chain, no interactions between the chains. So no folds, no bends, no nothing, which is not what we see here. We see a lot of bends. We see alpha helixes, right? So secondary structure is when you have alpha helixes. Okay, so alpha helix or beta pleated sheets. Okay, but these alpha helix or these beta pleated sheets are not interacting with each other. They're just simply existing, alpha helix and beta pleated. So there's no interaction in between. Okay? C, which is tertiary, is when you have alpha helixes and beta pleated sheets, or beta pleated sheets. So you have secondary structure. And on top of that, you have more interactions. So what are these kinds of interactions? Well, you're going to have like hydrophobic interactions, ionic interactions, sulfide bridges, and the tertiary structure is what we see here in this image, because we can see that everything is interacting together and folding together. So it's about the interactions, and it's about how everything's interacting together, folding together, binding together. The extra hydrogen bonding, the disulfide bridges, all of that. Okay, so this would be the correct answer here for the tertiary structure. The reason why it's not quaternary structure is because quaternary consists of multiple tertiary structures. And we don't have multiple tertiary structures, we just have one tertiary structure. And we can see that the alpha helixes, they all form just one protein. So if it had multiple polypeptide strands and each of them form their own kind of tertiary structure, then that would be quaternary structure. Okay, next question, 25. The graph shows the energy values during a chemical reaction. The horizontal lines represent the original energy values. I thought this question was really mean to put on a biology paper, but we'll go through it anyways. So the options are, which graph represents the energy changes once a suitable enzyme has been added? So once you add an enzyme, okay, B. So maybe this wasn't such a hard question for a bio student, but yeah, if you do chemistry, you'd know that an enzyme, what it does is it provides an alternative activation energy. And if you do bio, you know that enzymes catalyze reactions by lowering, well, quote unquote lowering, it doesn't really lower, but in bio, it's lowering the activation energy. So here we're looking for an activation energy that's lowered. So as you can see here on this graph, this is the original activation energy, the peak here. That's the energy that needs to reach before the reaction can progress, and therefore you can hit your reactants. Okay, and this will be your, before you can hit your products. This will be your reactants on this side, and this will be your product. So before you can reach your products, you have to hit that minimum energy requirement, your activation energy. So with a catalyst, that is lowered, and the only one that, or the only two that have a lowered activation energy are going to be B and C. So we can exclude A, and I can exclude D because they're showing the same activation energy. Now, what's the difference between graph B and C is the energy level of the products. So the energy level of the products should stay the same. Nothing else should be different except the activation energy, which is why B is correct, because C has a decreased energy requirement for the products, which is not going to change with a catalyst. Okay. 26, which part of the aerobic respiration directly involve oxygen molecules? B, yep, exactly, accepting electrons from the electron transport chain. So oxygen, yep, exactly. So the oxygen molecules are required for, are they required? So if this is my whole aerobic respiration process, I start with glycolysis, where glycolysis is anaerobic. That's why with anaerobic respiration, I also do glycolysis in the cytoplasm. That's why I can exclude A, because glycolysis doesn't need oxygen. After that, I have the link reaction, right? After that, I have the Krebs cycle and the electron transport chain. And all of those require oxygen. Now it says directly involves oxygen molecules. So these three steps, so B, C, and D, they have to be either relating to any of these three steps, and they have to be really exact and precise about where the oxygen is coming. Okay, so B says accepting electrons from the electron transport chain. So yes, absolutely, this is true, because remember, the oxygen is the final electron acceptor in the electron transport chain. Okay, so as you go down that electron transport chain, as the electrons build up, something needs to be there to grab them, and that will be the O2. So that's the electron transport chain. Oxidizing acetyl groups in the Krebs cycle. So an acetyl group is going to be your 2-carbon chain. Are we oxidizing the 2-carbon chain? No. We're breaking down pyruvate, which is a 3-carbon chain, and oxidizing the breakdown. So we're oxidizing that 3rd carbon. We're not oxidizing the acetyl group, which remains. We're oxidizing this one to make CO2. So therefore, that's why this one's incorrect. D says production of NAD from reduced NAD. No, it has nothing to do with NAD. It's just Krebs cycle, link reaction, and etc. Okay, the micrograph shows part of a chloroplast. Which reaction takes place in region B? Okay, so let's go through the question together. So in this chloroplast, first of all, where am I? So what are these? These are going to be my thylakoids, right? So these are my discs, my thylakoid discs. This is going to be my cytoplasm of uh, the chloroplast, basically, and that's called the stroma. Now, the thylakoids are green, and so they're going to contain a lot of chlorophyll, right? And so the thylakoids is the site of the light-dependent reactions in photosynthesis. The stroma is going to be the site of the light-independent reactions. Yep, see, exactly. Okay, so which reaction takes place in region B? It's going to have to do anything with the light-independent reactions. Anything that has to do with light, I'm going to ignore because that's going to take place in the thylakoids, the chloroplasts, and the photosystems and everything. Okay, so the photolysis of water molecules I'm going to exclude. C says generation of ATP by ATP synthase. So that happens during the light-dependent reactions. And so I'm going to remove C as well. D says reduction of NAD in photosystem 1. So the NAD gets reduced in photosystem 1. Again, it's photosystem. That's light dependent. So I'm going to exclude D, and so the answer is going to be A, carboxylation of ribulose bisphosphonate, because that's the only part of the light-independent reactions. Because what are the light-independent reactions? They are the Krebs cycle. And the Krebs cycle uses the enzyme Ribisco to add a carbon to the ribulose bisphosphate. Remember, we have this 4-carbon molecule, we add this, a 5-carbon molecule, we add this, it splits off immediately into 2 or 3-carbon molecules, and so on and so forth. Okay? So this question is a little bit confusing. Go back and review this topic, the photosynthesis one, because it's quite an easy question to answer if you know the content, if that makes sense. It doesn't require much application, which is great. So it's kind of a topic that if you understand well, you can get really, really good marks from that in the paper. Okay, 28 says the stem of a young plant is cut through, and the cut is cut through, and the cut end is is immersed in water containing a red dye. The plant continues to transpire as normal. In what region of the stem would the red color be visible soon after immersion? So it's saying that they cut open a plant and then they put it in this water, and this water had a bunch of red dye. So this water with this red dye, they're saying that after they cut off the stem, the transpiration still continues to take place. And so we now have a plant, and this plant is going to have both the xylem, and it's going to have phloem, and it has a lot of vasculature. And it's that vasculature that's going to be carrying this red dye above. So the red dye is going to travel up the plant, and it's asking what structure is it traveling up? Is it traveling up the phloem or the xylem? So the answer is going to be the xylem, because the xylem is going to be the site where the water transport is going to happen. And a really hint for that is it says visible soon after immersion. So the red dye will be there as soon as it was immersed. So the water would first go through the xylem. And obviously, I know the phloem would also have some of that water at some point because the water goes from the xylem into the phloem, but that happens later. So initially, the water goes up directly into the xylem because that's where the water transport is going to happen. And so the answer is going to be exactly D, for the reason that D shows the xylem, okay? And B is going to show you the phloem, which is not the correct answer because the phloem, it will have water, but it will have water later on, not directly after. Okay? Cool. And yeah, so what about 9? What is the benefit of having xylem vessels close to the phloem sieve tubes in plants? So we're having B. Yep. Cool. Anyone else? Yep, we've got D. Okay. Okay, let's have a look. What is the benefit of having xylem vessels close to the phloem? So this kind of requires an understanding of the transport in the phloem. So we know that sugars can flow in the phloem, and they do that, first of all, by the sugar gets loaded into the phloem companion cell, and then it gets loaded here. So now I have a bunch of sugars. In order for the sugars to travel, I need water from the xylem to then pass these sugars following what's we know as a hydrostatic pressure. So the water leaves the xylem and enters into the phloem using osmosis, really. And it does that using osmosis because there's going to be a lot of sugar, and water loves sugars, and so it will just enter through here. That establish a hydrostatic pressure gradient, and it will establish this hydrostatic pressure gradient, which means that it will go from an area of high pressure where there's a lot of water to an area of low pressure. So the hydrostatic pressure gradient allows the water to travel and to travel with the solutes of the sugar dissolved inside it. Let's go through options A, B, C, and D. So A says sugars can flow in both xylem vessels and phloem, and that's not true whatsoever, so that's an easy thing to rule out. Water can be supplied more easily to phloem. That kind of sounds like it would be true because that's why the xylem vessels being close to the phloem is really advantageous, right? A high concentration of solutes can be established at the source. So no, that has nothing to do with the xylem vessels. That has everything to do with the source and of the sugars and the companion cells. So we're going to exclude C. D says a hydrostatic pressure can be established to move sugars. Oh, but wait, it says to the source, not to the sink. And that's what we don't want. It's going to move it to the sink, in fact. So that's why also D is incorrect. And therefore the correct answer is B. And it does make sense. The fact that xylem is so close to the phloem means that water can be used in that transport of the sugars. So water can move across via osmosis easily. Okay. Question 30 has been removed for copyright reasons, and I really couldn't find this question, so we're gonna just skip it for today. What is a result of crossing over in meiosis? Question 31. We're nearly there. Got D. So remember that it's not asking what is a result of meiosis, it's asking what is a result of crossing over. Yep, perfect. So everyone's got this one. Everyone got D, which is exactly the true answer. So gene linkage has nothing to do with crossing over in prophase. It kind of does because we talk about how close the genes are and how likely they are to get recombined, and therefore how likely the genes are to be linked. But that's not really what we're kind of testing here. Non-disjunction, no, that's not a result of crossing over. That's a result of incorrect anaphase, right? Haploid cells, no, the actual meiosis results in haploid cells, but not specifically the crossing over. And the crossing over absolutely does provide variation in gametes because you create recombinant chromatids, and these chromatids all have a unique DNA that's different to the parent cell, different to the original parents. Okay. So in the fruit fly Drosophila, the alleles for red eyes and brown bodies are dominant to the alleles for white eyes and yellow bodies. Which cross would be suitable to determine whether the genes are linked? So for this, kind of take a little bit more time to solve it. So I'm going to give you guys a little bit more time, and I'm going to be writing the alleles. So the allele for red eyes and brown eyes are dominant to the alleles for white eyes and yellow bodies. So red eye, brown body are dominant. White eye is going to be small r, yellow body is going to be small y. Which cross would be suitable to determine whether the genes are linked? Well, you have to cross over flies that are going to have your dominant together and your recessive together, and see if the offspring are going to have only dominant or only recessive. Okay, so heterozygous red-eyed flies cross with yellow-bodied flies. So that's telling me I'm going to cross over a heterozygous red-eyed fly. So it's going to be RR, and so RR for red eyes and then brown bodies. So here I need to have, first of all, I need to have something that, because we're looking at genes are linked, I need to have a cross where I have two of those traits being crossed with two of those traits. Here it's telling me to cross big R, small r for the red eyes, X yellow-bodied, so small y. That's not going to help me if the genes are linked because that's crossing two different genes. So that's why I'm excluding A. White-eyed crossed with a homozygous brown body, the same exact reason. So white-eyed, so that's going to be small r, crossed with homozygous big B. That's not going to help me with a linked gene. I need to have the two traits in each parent. And so I'm going to exclude B. C says homozygous red-eyed, which is going to be big R, brown-bodied, big B, crossed with white-eyed, so white-eyed, so small y. Yeah, sorry, that was yellow-bodied. My bad. That should have been actually small b, but anyways, yellow-bodied and R. So this is what this is saying. So what would a cross like this reveal if how would a cross like this reveal if they are linked? So in terms of if these two were crossed together, because the traits are dominant, then these traits, if they were passed on to the offspring, I won't be able to tell if these traits are passed on because they're dominant or because they're linked. So the answer would not be C. Heterozygous red-eyed brown-bodied flies crossed with white-eyed yellow-bodied flies. So let's have a look at what they're trying to say. So big R, no, sorry, big R, big B, small b, crossed with white-eyed, so that will be R, X, yellow-bodied. So RR, yellow-bodied would be YY. So I don't know why I put YY for this trait. It should really be small b, small b. So I'm going to remove that, and yeah, I'm going to make this small b, because that makes more sense. I just got confused with the yellow. So there we go, small r homozygous and b homozygous. And in fact, this would be the correct answer because a cross between the homozygous will just give a dominant trait to all the offspring, and you won't be able to see if the genes are truly linked. Or to see if the genes are truly linked, you have to have the heterozygous with a test cross. It's the same as the crosses in your SL content, so only in the Punnett grid. You have to cross a heterozygous with a recessive in order to find the Mendelian ratio of 1 to 2 to 1, right? So you need to be able to do that. And here, and that's called a test cross. Here we're trying to emulate the same thing, where if I cross a heterozygote with a homozygous, that's supposed to give me a Mendelian ratio of 9 to 3 to 3 to 1, but it's not going to give me that if the genes are linked. Okay? So it's just a matter of remembering what genotype. It's a heterozygote and a test cross. So a heterozygote with a test cross. That's what it has to be in order to get the 9 to 3 to 3 to 1. And if it doesn't match the 9 to 3 to 3 to 1, then I know that the gene is linked. Okay? That's why the answer is D. But A and B you can exclude quite simply. Okay, does that make sense? Let me know if it doesn't. But the answer is indeed D, sorry. Okay, moving on to the next question. The graph shows how the frequency F of a trait within a population changes when subjected to selection pressures. Which graph shows stabilizing selection? So the dotted line is before selection, and the straight line or the continuous line is after selection. So it would be stabilizing. B, yep. Yep, absolutely. Exactly, it would be B. So let's go through it. So if this is before selection, which is the dotted line, and then this is after selection, what we've done is you've selected for the extremes, and that is known as disruptive. B shows stabilizing because you have the dotted line. And as you can see, this is the dotted line before selection, after selection. It's just stabilized to select for the middle characteristics. So the characters in the middle, the median. So this is definitely stabilizing. C is also not stabilizing because it's selecting for one extreme. And then D is also selecting for both extremes after selection. That's also not stabilizing. Okay. 34. What can occur? Oh, and sorry, I forgot to say that this will be directional. 34. What can occur as a result of exposure to an allergen such as pollen? Anyone else for D? Okay. So we can go through it kind of step by step. So when you're exposed to an allergen, the body recognizes that as an antigen. So it recognizes it as something that needs an immune reaction. And the way the body reacts is by having white blood cells. There's specific types of white blood cells, and they will release huge amounts of histamine. And this histamine is a molecule that triggers an inflammatory reaction. Okay? And this only happens if you are allergic to that molecule because you've already seen that molecule before, and therefore you trigger this really powerful immune response. So A says T lymphocytes produce antibodies. So A is just incorrect because it's never the T lymphocytes that produce the antibodies. Anyways, it's your plasma cells. And so A can be easily excluded. B says memory cells release antihistamine. No, the memory cells are there to redo an immune response and redo the immune response in response to a bacteria or a viral infection. And they're not there to release the antihistamines. The memory cells are going to be your memory T cells and your memory B cells, neither of which have a function of releasing antihistamine. Okay? C, the allergen acts directly on the blood vessels, causing vasodilation. So no. So the histamines that are produced, they are going to cause vasodilation because of the histamines, but the allergen doesn't directly act on the blood vessel. So that's incorrect. And the correct answer is in fact D, which is the white blood cells will release histamine. The release of the histamines will trigger inflammatory reaction, which part of that is going to be dilation of the blood vessels and making them leaky so that all the white blood cells can leave and enter into the site where the allergen is, but it's not going to be a direct process. Okay, nearly there, guys. We've got about 5 questions left. So 35 is what is fused with tumor cells in the production of monoclonal antibodies? Anyone else for C or a different one? So the answer is in fact C. So let's kind of revise the process of the production of monoclonal antibodies. So we will have antibodies that are extracted from a donor. So we will take the antibodies from them. And then we will, or I guess the code for the antibodies, the genes for the antibodies. And what we're going to do is we're going to take tumor cells because tumor cells are infinitely dividing. And therefore they can produce a lot of antibodies for us. We take those genes, we put them into the tumor cells, they fuse together to form something, and these will be like plasma cells, by the way. We're extracting everything from the plasma cell. So we fuse together a plasma cell, which possesses the genes to make an antibody, right? And that plasma cell is going to fuse with the tumor cell. So I'm going to actually color my plasma cell in green. It's going to fuse with the tumor cell, which is in orange, and it's going to create a hybrid cell known as a hybridoma. Okay, and this hybrid cell, which is a hybridoma, is going to have it's going to be possessing both the characteristics of a B cell, which means it's going to make the plasma cell, and it's going to possess the characteristic of a tumor cell, which is going to keep dividing infinitely and infinitely and infinitely. And so, indeed, this is the process. So it's asking, what is fused with tumor cells? So what's fusing with a tumor cell? It's going to be a plasma cell, and they will form a hybridoma cell. And so that's why we can exclude A, because it forms this, doesn't fuse with this. Antigens are excluded, specific antibodies are excluded because it's the plasma cell that possess the genes to make antibodies. And then they're going to make the antibodies afterwards, after the fusion and the formation of the hybridoma. Okay. 36, where do calcium ions that are released from the sarcoplasmic reticulum bind to stimulate muscle contraction? This requires remembering of the process for the muscle contraction. Yep, absolutely. It will be D. So if you guys remember, calcium is released from the, I'm going to draw a really, really bad sarcoplasmic reticulum, but this is the sarcoplasmic reticulum. Calcium is going to be released from the sarcoplasmic reticulum from the voltage-gated calcium channel. It will then go and interact with the muscle filaments, where it will basically bind to a molecule called troponin. So this is my muscle protein, right? And it will bind to a protein called troponin. So this is my muscle. Let me just draw it in a better way. So this is my muscle here, and this is my protein troponin. It will bind to troponin, and when it binds to troponin, it causes the cell, so the calcium will bind to this, it causes the tropomyosin to move out of the way. So when it binds to troponin, it will cause the tropomyosin to move out of the way, and it will reveal the binding sites on the actin. Sorry. It will reveal the binding sites on the actin that then the myosin head can bind to. So remember, calcium will come and it will bind to your troponin. When it binds to the troponin, that will move the tropomyosin. So the tropomyosin is going to be the little thread that moves, or the little filament that moves. and the troponin is the binding site for the calcium. Okay, cool. And these obviously it will expose the binding sites for the myosin, sorry. Cool. Last few questions. The diagram below shows the bones, muscles, and tendons of the elbow joint. Which muscle is the extensor, and to which bone is it attached? Okay, so yeah, we already have a D in the chat. Oh no, that was a previous D. Have a go trying to answer this one. Okay. Yeah, this one is also D indeed. So first of all, let's go through the question like bit by bit. So we have the elbow joint. So which muscle is the extensor? So what movement is extensor? Remember that flexion is reducing the angle between this bone and this bone. Extension is increasing the angle at the elbow joint. So this is extension. For this extension, this is my tricep. So my tricep is contracting. So I can exclude A and B based off of that because it's not my bicep. And now it's about what is the site of attachment for this muscle. So is it going to be the ulna or the radius? So remember that the radius is going to be this bone here, and it's the side of your thumb. That's going to be the site of attachment for your bicep. So if you put your arm this way, you can kind of like, this is the thumb, this is going to be my radius, it's going to be for my bicep. And my ulna, which is about my pinky finger here, is going to be for my tricep. Okay, so that's the way you can remember that. So that will be the answer D. Cool. Next question. 38. Where is the greatest quantity of water reabsorbed from the nephron? You've got C. Any other answers? Okay, got another C. Okay. So let's go through it bit by bit. So water reabsorbed from the nephron. So it's not water entering the nephron, it's water being taken away from the nephron. So at the Bowman's capsule, water enters the nephron. Okay. And as water enters the nephron, from the blood, so from the arterioles into it's got like these convoluted vessels, and then the Bowman's capsule, water enters that way. So H2O goes that way, and it goes then into the PCT. Now, at your PCT, or your proximal convoluted tubule, this is where the majority of the water reabsorption will take place, actually. About 80% of the water will be reabsorbed here. And if you remember, it's because of the fact that all of my sugars are being reabsorbed here as well. So my sugars, my amino acids, majority of them, a lot of materials are being reabsorbed at the PCT. Okay, and so the sugars, as they get reabsorbed into the blood, so they go out, the sugars will go out, the water will simply just follow them. Okay, the water will simply just follow them via osmosis, and that's why 80% of the water reabsorption happens here. Now, in the loop of Henle, most of the osmoregulation happens here. So the collecting duct and loop of Henle, this is where osmoregulation happens. So remember before I told you guys, be careful, because you might be tempted, if a question asks explain osmoregulation, you might be tempted to include the fact that the PCT reabsorb all of the water, the 80% of the water. But that's not osmoregulation, that's just reabsorption. It's passive. It's just, it goes via osmosis. It's not being actively controlled. Osmoregulation is when you're actively controlling the water content. And that's through active transport mechanisms that are done in the loop of Henle and in the collecting duct. So loop of Henle is after the PCT, and we have the loop. And remember, it's very, very salty inside the loop, and we have the descending loop and the thick ascending loop. And the thick ascending loop is permeable to salts but not water, and the thin ascending loop is permeable to water and not salts, and all of that. That's osmoregulation. And even though the majority of water doesn't get reabsorbed here, it's actually the majority happens in the PCT. This is where osmoregulation takes place. And osmoregulation will take place here and in your DCT after the loop of Henle. And then, not in the DCT, sorry, that's like again reabsorption, but after the loop of Henle, it's going to take place in the collecting duct. Okay, so osmoregulation takes place in these two. However, the majority of the water gets reabsorbed in the PCT. Okay, does that make sense? just because water comes out following the sugars, following the amino acids and everything. But do remember that osmoregulation will not take place at the PCT because osmoregulation is using actual ATP in order to regulate the amount of water. And that's going to be in the Lupini and the collecting duct. Cool. 39. At what stage of spermatogenesis does the first division of meiosis occur? So this is kind of, if you know it, you know it. Otherwise, it might be a little bit confusing. Obviously, we know that the spermatid, after division, will mature to form the sperm cell. And that has nothing to do with meiosis, so I can exclude that. Secondary spermatocyte to spermatid, that's probably not the first division of meiosis. That's probably the second division of meiosis because that's the last division. So it's kind of between A and B. Anyone have any guess on whether it's A or B, or if someone knows? Okay, so actually I'm going to use an image that I found earlier. So this is the spermatogenesis process. So as you can see, this is my spermatogonium. And when it's a spermatogonium, it's just an early kind of stem cell, really. And in order for it to actually start going into meiosis, it has to first turn into what's known as a spermatocyte. So spermatocyte is when it actually starts dividing. So spermatogonium is kind of an early like stem cell. It's quite differentiated, but it's still growing and it's still preparing. As soon as it's growing and preparing, and then it's finally now grown enough, it's going to be called a spermatocyte. When it's a spermatocyte, then it does its division. When it's a secondary spermatocyte, it will do another division. Okay, so the word spermatocyte is when the divisions happen, really. So going from a primary spermatocyte to a secondary spermatocyte, that will be your first division. Okay, and therefore the answer is going to be B. A is not going to be the time of the first division because the spermatogonium will not divide. It's only the spermatocytes that will undergo meiosis. Okay, and this will be your second division for spermatocyte. So the spermatocyte is what's dividing. Cool. Which statement applies to the placenta? And it's the last question. I'm just going to move this image out of the way. A. Okay. B, C, or D. Any other answers? Okay, so the answer is indeed going to be A, and let's have a look at the reason why. So B says maternal and fetal blood mix at the placenta. And that's not true. The maternal and the fetal blood don't mix. In fact, the fetal blood, so this is the placenta, or the fetal blood comes in via blood vessels. Now these blood vessels are open at this end. So they're open, and the mother's blood vessels come in here, and they're open at this end. So we call them open-ended arterioles. Okay, so the mother is supplying a bunch of blood, and there's a place called the chorionic villi, where the mother ends up supplying blood and the fetus supplies blood. But the mother's blood never mixes with the fetus blood. If it does mix, then the mother's blood will enter the fetus, and that's not good. And vice versa, the fetus blood will enter the mother, and that doesn't happen. It doesn't mix. And at this place where the chorionic villi is, there's kind of like a divide, a tiny, like it's almost an invisible divide where the mother's blood enters and then the fetus blood here, and there's no actual mixing. And in fact, the carbon dioxide from the fetal side will actually diffuse into the mother side of that. And that's the gas exchange. The CO2 will come here as a waste. The mother will be able to excrete that waste. And the O2 will diffuse again to the fetus side just because the fetus side is able to trap the oxygen better. And therefore that also happens via diffusion. So the blood doesn't mix at the placenta. It's just that the gases diffuse. Okay, so that's why A is correct and B is incorrect. If an ovum is not fertilized, the placenta is lost during menstruation. No, it's not the placenta that's lost. It's the endometrium that's lost in the menstruation. The placenta is lost, and that's a miscarriage. Endometrium. So if the placenta is lost, it's a miscarriage, but this is just menstruation normally. So it's just the endometrium lining, and that's if the progesterone levels drop. The umbilical cord connects the placenta to the mother's abdomen. No, it connects the embryo. So it connects the actual fetus. The embryo, it's got all of the blood vessels and it connects it to the mother, but that's not true. And so therefore A is the only correct option. Okay. So that's the end of the paper. Congrats on staying this long, guys. We finished the whole paper 1. If anyone has any questions on any of the content covered today, just let me know. And if you encounter any difficult questions in your past paper revision, you're always welcome to email me. But yeah, that's it for today. Thank you so much for sticking by. You're free to go now. And yeah, sorry we had to run over by 15 minutes.